This is part 2 of Chapter 8's lecture. So we left off discussing endergonic and exergonic reactions. So energy is released from the downhill reaction of a catabolic pathway, and it can be stored and then used to drive an uphill reaction. The size of the delta G for an exergonic reaction is defined as the maximum amount of work that the reaction can perform. So the greater the decrease in free energy, the greater the amount of work that can be done. So the greater this difference between our starting reactants to our products, the more that there's an ability to do work. Delta G is a change in free energy, the ability to do work. So an example of this is with cellular respiration. We have the reaction of C6H12O6 combining with 6 oxygens, releasing 6 carbon dioxides and waters, and energy. And that change in energy is a negative 686 kilocalories per mole. So that's the delta G of the chemical reaction of breaking apart a sugar molecule in cellular respiration. Another way to say that is that for every mole of glucose, 180 grams of glucose, that you break down by respiration, there are 686 kilocalories of energy available to do work in a cell. So the products have 686 less free energy kilocals per mole than the reactants started out with. So the products being the CO2 and the H2O, they have 686 kilocalories less free energy than the C6H12O6 and oxygen. So this is an exergonic reaction that is producing this 686 delta G kilocals. In contrast, we have the endergonic reactions that have to absorb free energy from the surroundings. And endergonic reactions have a delta G that's going to be positive because we're going from one energy level to a higher level. Endergonic reactions are not spontaneous, and how big the delta G is the quantity of energy that's needed to drive the reaction. So if in cellular respiration we have 686 kilocalories per mole released, then you would think that photosynthesis, which is the reverse reaction, must require an equivalent amount of energy to be invested for it to occur. And in reality, that is exactly the case. So the delta G here, if you were to be measuring photosynthesis and the anabolism, the anabolic reaction of making a sugar molecule, it would have a delta G of plus 686 kilocalories per mole. And photosynthesis is a very endergonic reaction that has to be powered by an absorption of the very strong energy, light energy, from sunlight. In an isolated system, in a closed system, reactions are eventually going to reach equilibrium and no longer be able to do work. And so if that occurs in a cell, which is fortunately not in an isolated system, if a cell reaches metabolic equilibrium with a delta G of 0, that means it's no longer doing any work, and it also means it's a dead cell. So one of the defining features of life is having metabolic disequilibrium. So not having metabolic equilibrium is important as a definition of a living cell. Cells are able to keep disequilibrium because they are open systems. So there's a constant flow of materials into and out of the cell that keeps the metabolic pathways from ever reaching equilibrium. And that way a cell is able to do work throughout its life. This is another way to graphically look at delta G in both endergonic and exergonic reactions. So instead of looking at that line graph with the different curves, you can look at it as a bar graph here. And we can see the reactants have this amount of free energy and the products have this amount of free energy. This decrease in the amount of energy is a negative delta G. So that tells us this is an exergonic reaction. Energy has been released. It's a spontaneous reaction. And here, if we have reactants with this low free energy and the products have a higher level of free energy, that's telling us we have a positive delta G, the difference between these. It's going to require an input of energy, making it endergonic, and it will be non-spontaneous. So now we're going to take a little side note or side trip here on discussing ATP, because ATP is a way that cells get energy to run their exergonic and endergonic reactions. So a cell can do three types of work, primarily chemical work, transportation, or mechanical work like movement. And so in order to do this, the cells have to manage their energy resources and materials that are available by coupling reactions, using an exergonic process to drive an endergonic one. And most of the energy coupling in cells is accomplished by using ATP and its interaction movement to ADP and then back again. So this is a way that we're coupling an exergonic and endergonic process. A reminder of the structure of ATP is on this slide. It's a nucleotide triphosphate. It has the sugar ribose, and its nitrogenous base is adenine, and it has a chain of 3 phosphate groups attached to it. The bonds between these phosphate groups can be broken by hydrolysis, and when the last one, the terminal phosphate bond, is broken, you get a molecule of inorganic phosphate released. You also, this is an exergonic reaction, you get a release of energy. And under standard conditions, the delta G for this removal is a negative 7.3 kilocalories per mole. What's important to note, something a little bit surprising, is that the release of energy in this exergonic reaction is not actually from breaking the phosphate bonds, because those are actually weak covalent bonds. But actually what's occurring here, the energy from the exergonic reaction of ATP hydrolysis is actually that delta G is the energy that is actually being used by the cell to do work with the help of ATP's hydrolysis. So again, when the terminal phosphate bond is broken, this is another picture showing that, there's a molecule of inorganic phosphate that leaves ATP, and then the ATP is then ADP, or adenosine diphosphate. And under normal conditions, there's that negative 7.3 kilocalories per mole in that delta G from ATP to ADP. So the coupling of a reaction is what is depicted here of an endergonic and exergonic reaction, because what we're doing here is building this molecule here, which is going to be an endergonic reaction. So in order to combine these two molecules into this one, we need an input of 3.4 kilocalories per mole. So it's positive delta G here. And so we use ATP to do that. And so there's a removal of that phosphate group, which is then going to release a 7.3 kilocalories per mole. And if we add those up together, we end up with a net delta G of negative 3.9 kilocalories per mole. So what we have occurring here, we have an actual, this reaction right here is actually endergonic. But for the whole system, we actually have an exergonic reaction. So in this way, we're able to end up, remember that to be exergonic means it's going to occur spontaneously. So by coupling these two reactions together, we're able to transform this endergonic reaction that is non-spontaneous into one that is spontaneous. So if reactions are downhill, why aren't they just happening spontaneously? They actually can occur without an input of energy, although they usually are not going to occur very quickly. There are some spontaneous processes that don't require much energy, like an explosion can be instantaneous. But then there are other reactions that are spontaneous, like the rusting of an old car. It can take years. So a spontaneous process is one that is energetically favorable, but it may not occur on its own. Any process that cannot, or it actually is going to occur on its own, but it's going to be very slow. But any process that cannot occur on its own is actually called non-spontaneous and will only happen if we add energy to the system. So the energy that we use, for example, to start a fire, adding a spark, is activation energy. So even breaking down a large molecule into smaller ones requires an input of energy. And that's because large biomolecules have stable bonds. And so they have to absorb some energy to break those bonds. A lot of times activation energy, we think of it as only being associated with enzymes. But actually, activation energy can be supplied in the form of thermal energy, and so that the reactant molecules absorb that thermal energy from the surroundings, which accelerates the reactant molecules so that they collide more often with more force. Or they can, if you're trying to break apart a molecule, having a catabolic reaction, you can agitate the atoms within the molecules by heating the system, adding thermal energy, so that making the breakage of bonds more likely. And when the molecules have absorbed enough energy for the bonds to break, the reactants are in an unstable condition called the transition state, so that they can then be broken. The only problem with this whole setup is that simply adding heat to get a reaction to occur, to actually provide the activation energy, becomes a problem for cells. Because if too much heat is added to cells, we have the denaturing of proteins and the actual killing of a cell. So there has to be another solution to supply the activation energy needed for a reaction to occur. And that's where catalysts come in. So catalysts reduce the amount of energy, that activation energy, that's actually needed to start the reaction. You can see that picture. This is the classic picture of here's the normal amount of activation energy needed to go from this state to this one. But with the enzyme, it's lowered this amount here. So with the catalyzed reaction. And actually, I just stated what are our biologic catalysts, which are the enzymes. I want to remind you, or let you know if you haven't heard this before, that the symbol for this activation energy is a capital E with a um,